So pedigree analysis. So we have 4 laws, right? So law of unit pair. So Mendel laws are law of unit pair of alleles or factors. So law of dominance and recessive, law of segregation. So law of dominant and recessive. Law of dominant and recessive. So law of independent assortment. So law of independent assortment. So these are all the laws which related to the inheritance proposed by George Gregor Mendel. So George Gregor. So George Gregor Mendel. So George Gregor Mendel, he was called as a father of genetics. Why? Because he has given the various laws associated with inheritance pattern from parents to offspring. So inheritance of the traits, inheritance of the characters. So this is the basis for modern genetics. Okay, that's why he called as a father of modern genetics. So he did experiment on the garden pea plants. So garden pea plants, and he proposed, so one unit pair is responsible. Okay, and they follow the law of segregation, law of dominant and recessive, law of independent assortment. So these things, though he did experiment in the garden pea plants, but his validations are accurately true when it comes to all the species, such as plants as well as the animals. So those laws, when they observed in human being in the study of pedigree analysis, determination of the blood groups, determination of the sex of offspring. So determination of sex of offspring. So sex of the offsprings. Next, so medico legal cases are RH incompatibility also. So yesterday I didn't discuss about RH incompatibility, RH factor incompatibility. So today we'll discuss. So next one, medico legal cases. So medico legal cases such as paternity disputes, missing persons, personal identification, exchange of the babies in the hospitals. So medico legal cases, medical applications. So medical applications means identification of the various diseases and their characteristics and the type of inheritance. So dominant diseases, how severe, recessive genes are how severe. So this is how sex-linked diseases. And seventh one is, so genetic counseling. So genetic counseling means here in a family, for suppose in a family from generation onwards. So if you go to the dominant disease, so dominant disease, so genetically autosomal dominant or sex-linked dominant. So dominant disease means it expressed or it exists in every generation. So every generation dominant traits are present. Understand? So in a family, those people are dying because of this disease in every generation. Okay, and one of the person born in that family, so he has suspicious about whether this person is affected with the disease or not. So that kind of doubtfulness. And what would happen if he get married and produce the offspring? So first of all, doubts related to whether that himself or herself affected by this disease or not. Whether if he affected, can he get married or can she get married to produce a progeny? So these kind of advice they need it. So these persons we call as a counsel. So counselor. So those person, that is geneticists or medicine man, who is studying about whether the person is affected with the disease or giving the advice about getting the children or not. So that person we call as a counselor. So counseling, who is seeking the advice from the counselor, we call as a this suspicious person, or probe and prosita also we can call it. So that means many diseases are there. They are fatal or severe in nature. So in a family, if these diseases are present, can any person get or not? Or can he get married and produce the children? These kind of answers are seeking that we call as a genetic counseling. Okay. So genetic counseling here, the counselor identify that whether a person is affected, carrier, or it is passed to the next generation based on the nature of the disorder. So a disorder is a autosomal dominant, autosomal recessive, or X-linked dominant, X-linked recessive, or Y chromosome disorder. So these are all the things. Because he has the knowledge about the Mendelian laws, so he will apply and he can predict that whether the person is affected or not. So this is called genetic counseling. Genetic counseling itself as a 10 marker or 15 marker question that we will deal separately. So eighth one. So creating hybrid varieties. So hybrid varieties such as fruits, vegetables, and animals. So fruits, vegetables, and animals. So disease resistancy. Okay. High yielding varieties. And early maturing. So early maturing. So these are all the favorable traits for economy. So if we rear an animal, so they give more meat, more milk, and early they mature and early they produce, and they have least diseases affected, then these are all economically viable. Understand? So we will experiment. We will take these favorable traits from the different animals, and we create a hybrid varieties. Hybrid varieties, we will get all these three benefits in single species. So because these traits are governed by genes, and these genes are followed the Mendelian laws, we will cross fertilize all these species, and we will brought a hybrid variety. So hybrid variety. This is another one. So these are all the some kind of applications of Mendelian laws. So in additional supplementary, if you want, right, we can understand the nature of the diseases. So gene nature. So nature and pattern of inheritance. So pattern of inheritance of the genes. So this is how you have to write all these things. So where we are using the Mendelian laws in human family study means all these areas we will use. So generally if it is 20 marker question. So this is the continuation of the yesterday one, because we had discussed most of the things. So today we are discussing about limitation of Mendelian laws in human family. So limitations of Mendel laws in human family. So because he was a very clever, so he was a monk. So because in his courtyard, so he created the hybrid varieties of garden, so by choosing the various characteristics. So garden pea plants. So he has chosen contrasting characters such as one is tall, another one is dwarf or short. So these, when they cross fertilized or hybridized, all the things they come into the tall. So these are all the progenies of first. So progeny of these hybridization, and the plants born in these generations are hundreds of plants, and within the 1 month or 2 months, again they are ready for the ripening. In the ripening time, again they cross fertilize between the talls. So number of talls are produced. So self-fertilization among the talls, which are all the first generation from the hybridization. So these self-fertilization led to the again tall and dwarf appeared, tall and short appeared. So why? Because it is convenient. So the tree hybridization, it takes very less time. So tree hybridization at maximum, garden pea plant means how many months it will take to get the flowering, okay, seeds coming, and again fertilization. So it's like 3 months, 2 months at maximum, we can do this kind of fertilization. So that means the experiment conducting and the results obtaining, it will take very quick time. So very quick time in plants, because plants are seasonal in nature. In maximum 3 to 4 months, the crop also get over. But what about the human beings? So human beings, if two contrasting characteristics, per se one male, another female. So male is a taster, female is a non-taster. So if they get married, then what would happen? How many progeny they can produce? Hardly 4 to 5 progeny. And so what would be happened when this 4 to 5 progeny to be grown and grown into adolescent? After that, they became fertile and they should get married and they produce the children. So that means this is the first generation of this mating. And when next generation comes after 20 or 25 years, such a vast time, we have to observe, okay, whether Mendelian laws are applicable in the human family or not. Second thing, so there is no moral ground. So garden pea plants, tall and short, they cross fertilized. All the next generation are tall. When these tall are again cross fertilized, tall, short again appeared. But here, all these children born for this couple in the next generation are brothers and sisters. So brothers and sisters. That means mating between the brothers and sisters are prohibited in human being on the moral grounds. So moral grounds. Understand? So progeny wise, very less number of progenies are born in the human beings. Second thing is self-fertilization or cross-fertilization between the siblings is not allowed in human families. And when we want to ascertain whether these rules are exactly working or not, we have to wait like 20-25 years. Understand? So these are all the limitations. So plant lifespan itself is like 3 to 4 months. So within the 1 year, we can verify that all the Mendelian laws are properly working or not. So the same, if for one of the trait, when we have to verify in human being, so it is very complicated. So time wise, on moral ground, okay, so human behaviors are influenced by the environment and so many factors. Understand? So this is how Mendelian laws, though across the generation we believed and we documented they are working fine in the human being, but experimentation to be proved, it's very difficult. Okay, it's very difficult whether the laws are operating or not. So what are all the problems of here in man family? The problems are many. First one is limited family size. So limited family size. So the contrasting characteristics pair, they produce only limited number of children. But here unlimited, so look, hundreds, thousands of progenies are born in the hybrid varieties. Okay, second thing is, so you can say growth and sexual maturity. So growth and sexual maturity of the offspring takes a lot of time. So large period. Here it is in 2 months they can grow, again ready for the cross fertilization. Third one, so experimentation prohibited. So experimentation prohibited. So between the siblings. And fourth one, so plant inheritance is simple. Plant inheritance is simple. But in human it is complex. But human complex inheritance. So if the examiner wanted test you, so could we apply Mendel laws in human beings for experimentation assertion? Yes, we can do, but there are some problems. Okay, so generally what he did is Mendel, First generation, they produced a progeny. Okay, so self-pollinated or cross-fertilization among the progeny that led to the third generation. So this is the parental generation, F1 generation, F2 generation. So F1 is the result of the parental generation. F2 was the result of the mating among the F1 generation. So in a human being, this F1 generation individual constitute brothers and sisters. So mating among them prohibited every region. Understand? So in plants, it is a tall pea plant, short pea plant. Okay, they produce like hundreds of tall plants. So nobody prevented the Mendel to do experiment. Why? Because it is a plant. Understand? But in human beings, it is difficult. So this is how some complications are there. Though we can do it, but some complications are there applying Mendelian laws in human being. But still, they are well documented based on the instances available. Understand? If one of the person is like, how did they manage it? If one of the person is A blood group, another person is B blood group. Okay, so their children are born like A blood group people. Okay, so AO, BO, AB, okay, OO. So this is the genotypes. So these are all brothers and sisters. So these are all brothers and sisters. So why? Because they are born from the same couple. So here OO, if they mate with another person from another family, instead of in the progeny or siblings, we are taking the case from another family. So this is how multiple cases they derive, and they ascertain that whether it is working fine or not. So this is the application of Mendel laws through available instances they documented. So they documented for the blood groups, antigens, antibodies, and everything. So now we are moving into this is not that much important for the exam, but you should remember, so it is not that much easy to experiment on trees same manner in the human beings. It is very complicated. So now we will see exceptions to Mendelian laws. So exception to Mendelian laws. Exceptions to Mendel laws. So what do you mean by Mendel laws? First of all, you should see an individual is there. He has 23 pairs of chromosomes. So now loci is there. Each one is a loci. Here two alleles are present. So these are all we called as a alleles, and each loci we referred as a gene. So this is the gene. Each gene has two alleles, allele 1, allele 2. So both alleles are same, like AA. So that means AA means on the both chromosome, two alleles are same. Or in another case, AO. One allele is A, another allele is O. So if a loci or gene, both are same. Loci term or gene term, both are same. So loci has two alleles or gene has two alleles. So that means the points which are all present on the chromosomes we call as a alleles. For unit pair, it is required called as a gene or loci. It is required for the production of any product, that is protein synthesis. Understand? So now you can see, so this is how genes are arranged on the chromosomes. So gene is nothing but a loci. So this is arranged on the chromosomes. So like this, genes are arranged. So these are look like a beads on the string, according to Morgan theory. So each loci produces one product or one protein. Understand? So generally, each loci or each gene produce one product. Each gene produces like one product. The best example is nothing but chromosomal pair 9. One of the loci produces blood groups. So chromosomal pair 9. So chromosomal pair 9. So here one of the loci which is responsible for production of antigen on the RBC cell surface. So if this loci has three alleles. So A allele, B allele, O allele. If this loci has AA allele, here antigen is A. So antigen is nothing but a protein produced by this loci. If this loci has A allele, here the antigen antigen variety is A antigen. Here the loci is BB. The antigen is a B variety. Here the loci is OO. No antigen produced on the cell surface. Understand? Here the antigen is like one allele is A, another allele is B. Here antigen A and antigen B both are present. Antigen A, antigen B both are present on the RBC cell surface. Understand? That means loci is giving output in the form of antigen. Antigen is nothing but a protein. Understand? So that means every loci purpose, every gene purpose is nothing but to produce product that we call as a protein. That we call as a protein or protein synthesis. Proteins are in the form of hormones, enzymes, okay, antigens, antibodies. Okay, these are all the products of protein. So which are all produced by any one loci. So now the modern genetics or modern DNA decoding come to know that 22,000 genes are there. So 22,000 genes are there. So gene means locis. 22,000 genes are there. Okay. So 22,000 locis, they collectively produce 96,000 products. So 96,000 products. So that means here some of the genes, a single loci produces one output. Sometimes 2-3 locis collectively produce one output. So if every one loci produces one output, it could not be possible 96,000. Understand? So some of the locis, a single loci gives a single output. Sometimes multiple locis together give single output. Sometimes a single loci give multiple outputs. Sometimes a loci output is controlled by another loci. Understand? That means though genes are present on the chromosomes, all genes are not functioning in the similar in manner. So some are regulatory gene. So this gene loci function is controlled by this loci that we call as a regulatory genes. Okay, so regulatory genes. If single loci produces multiple outputs, that we call as a pleiotropy genes. So pleiotropic genes. Understand? A single loci producing single output, that we call as a Mendelian loci. So Mendelian locis are nothing but single loci, single output. Single output. Understand? So this is how genes are further classified. So though we know the Mendelian laws are 4, these Mendelian laws are not working on all the locis, all the genes. So which type of genes are qualified? So those loci which produce a single output or single character, single trait, that locis are suitable for Mendelian laws applicability. So the pleiotropic genes, regulatory genes, or remaining classifications are there. That also I will give. These are all the genes never follow the Mendelian laws. Understand? So then how many Mendelian locis are there in total DNA or 46 chromosomes? For suppose only 2 to 3,000 genes are. 2 to 3000 genes are, they produce one low key, one output. Okay, here law of dominant and recessive applicable. So law of dominant and recessive applicable only 2 to 3000 low keys, and these low keys are Mendelian low keys. Remaining low keys are regulatory low keys, pleiotropic low keys, or epistaticism. So another kind of epistaticism also there. Epistatic genes. So epistatic genes. So there are multiple varieties are there, out of which we are talking about only Mendelian genes. Mendelian genes are single low key, single output, or single character. If a low key giving multiple outputs, it's not the Mendelian law. This low key never follow Mendelian law of dominance and recessive. If a low key give only one output, so for suppose if you take chromosome 9th pair, this low key is only producing the blood groups. Okay. So in another case, a low key producing like X product, Y product, Z product. 3 products are coming from the single low key. If you assuming that blood group production is one character, that is X, that low key is always producing X. Understand? So within the X varieties are there. But if a low key produces like X output, Y output, Z output, that low key we call as a one low key, multiple outputs. So how these 96,000 outputs are coming from the 22,000? So if you take that, out of this 22, 3,000 genes are single output giving. Single output giving. That means if you remove the 3,000 from here, 93,000 are there. So 93,000 outputs are produced by 19,000 genes. Okay, so that means 3,000 genes are responsible for 3,000 outputs. You reduce 3,000 from here, total 93,000. You reduce 3,000 genes from over here, 19,000. So 19,000 genes are giving 93 outputs, means on average, approximately single low key producing 4 type of outputs. 4 types of outputs. Understand? So in these cases, Mendelian laws are not applicable. So in our total DNA, for example, 19,000 genes are not following the Mendelian rules. Not following the Mendelian rules. Only 19,000, 2 to 3,000 genes are following Mendelian laws. So if a low key is following Mendelian laws means what? So if a low key is following Mendelian laws means the low key must have applicable this character. So law of dominance and recessive. If a low key has different alleles, if a low key has different alleles, so same alleles means AA, same allele. Different allele means AO, AB, okay, so BO. These are all the different combination. If a low key has different alleles, it must have law of dominance and recessive. So law of dominance and recessive. A low key, if it is following law of dominance and recessive in heterozygous situation, that low key we call as a Mendelian low key. That low key we call as a Mendelian low key. Understand? So Mendelian low key means this is how you should remember. A low key, whether it is a Mendelian or not, means whether that low key, when it has heterozygous condition, it is following law of dominance or recessive, and that low key producing single output or not. This is the 2 conditions you should remember to classifying whether the low key is a Mendelian low key or not. So remaining low keys are not qualified under Mendelian laws. Why? Because they produce multiple outputs. Okay, they act as a regulatory gene. Instead of producing any output, they act as a gene interaction. Okay, so they act as, okay, don't following the law of dominance and recessive. So non-Mendelian low keys. So non-Mendelian low keys. So without producing output. So without producing output, but control the expression. Control the expression of another low key. Another low key. So this we call as a gene interaction. So gene interaction. For suppose, so if you take two chromosomal pair, here one loci is there, here another loci is there. So this loci function is dependent upon this loci working. That means this loci never produce any output, but its role is it control the expression of the first loci. Okay, here without giving any output, but it can control the output of another one. So it is called, that means the dependency of the first loci output is dependent on the second loci. This we call as a gene interaction. Understand? So it means whatever the genes here present, nothing to do without involvement of the second loci. In this case, Mendelian laws is difficult to operate. Understand? So the second one is some genes producing multiple outputs. Multiple outputs. So multiple outputs. So why you can see if in a Mendelian case, one person is a taster, another person is a non-taster. So what about the second generation, F1 generation people? F1 generation people all are born as a tasters. Okay, what about that F2 generation? F2 generation people are two types, tasters and non-tasters. Tasters and non-tasters. Understand? So now, in any generation, the character produced is one of the character of the parents. So taster, it is present in the parents. Non-taster, present in the parent. Taster, present in the parent. That means in every generation, whatever the character it is appeared, it is coming from the parents because it is present on the parents. If a loci has multiple outputs, per se, parents produced X output. Okay, one of the parent produced X output, another parent produced the Y output for the same loci. Same loci. One output is X in one parent, another parent it is Y. Why? Because the gene has the capability of producing multiple outputs. Now, so when they marry and they produce the children, children have Z, A, B outputs. So now, whether X is inherited or not, we cannot know. Why? Because these are also output of that loci. So that means if taster is inherited or not, depend on the Mendelian laws. If X is not coming in the next generation, and in the next generation also that loci is not giving the X, whether Mendelian laws operating or not, we cannot ascertain. Understand? And third condition is, so the loci must follow, or loci must not follow law of dominance and recessive. Law of dominance and recessive. So all the locis fall under any one of this category are non-Mendelian locis. Non-Mendelian locis are genes, they are more than 90,000 in human DNA. Okay, opposite. If a single loci producing single output. Okay, so next law of dominance and recessive. So these locis we call as a Mendelian locis. So in our human DNA, those are all present 2 to 3,000 genes. So they produce single output. So this is the categorization of Mendelian traits, non-Mendelian traits. Understand? So now we are going to the exceptions. Exception discussion. So exceptions to Mendelian laws. So exceptions to Mendelian laws. The first one is epistatic loci. So epistatic locis never follow Mendelian laws. What do you mean by epistatic loci? So generally, chromosomal 9th pair, one loci is responsible for the blood group formation. So blood group formation. So here, if it is AA, the blood group is A. So it is AO, blood group is A. So BO, blood group is B. BB, blood group is B. Okay. So AB, blood group is AB. OO, blood group is O. So these are all the combination. So independently, this low key based on the combination, it gives the output. So no other gene controls. But in the DNA, there is one low key. Here, QT alleles are there. QT alleles. So if this QT alleles in this low key, both alleles are QT. Both alleles are QT or homozygous condition. Homozygous condition. Both alleles in the same loci are QT alleles. In this case, it influences the function of this one. Okay. If both AA, the antigen should be A. If QT alleles are in homozygous condition, it stops the formation of antigen. So no antigen present. So even though your combination is AA, because RBC cell surface has no antigen, the output of that person is O blood group. Output of that person is O blood group. Understand? So AA person output must be A blood group. Because here QT alleles are there. This low key prevented formation of any antigen on the RBC cell surface. Absence of antigens on the RBC cell surface is nothing but a O blood group. So this kind of combination we call as a Bombay blood group. So Bombay blood group. So what is the problem here? What is the problem here? So on the low key, AA means the person should have A blood group. Okay. So then the person received the blood from A people or O people. O people. But the person is showing O blood group. The person is showing O blood group. From whom he will receive the blood? From whom they will receive the blood? So this is how the case is like why it was popular. So mother has O blood group. Father has O blood group. So O blood group. Then what is the children blood group? Generally, all the children should have O blood group. So all the children should have O blood group. But the children are born with A blood group. Children are born with A blood group. How it is possible? Because output of the female is O blood group. But what is the genotype? AA. What is the genotype here? OO. Understand? So that means in this combination, all the children are born with AO because A is dominant. So all the children are expressed A blood group. Understand? So it means impossible thing. The doctor was shocked. So both parents are O blood group. How could it is possible that blood group of child is A? They understood that the mother have AA. Okay, but mother output O blood group is coming because its antigen formation is controlled by the QT alleles. If QT alleles are in single dose or QT alleles are absent, the mother must have A antigen. Because of presence of QT allele, she has no antigen and she shown as a O blood group. But our genotypes are AA. It is passed to the children and O alleles are coming from the father. The children are A blood group. So this is how. If there is no QT allele, so here this loci can easily form the antigens. If QT alleles are there, this loci cannot able to form the antigen. So that means if no QT allele, this loci act as a Mendelian loci. Mendelian loci. If QT alleles are present in homozygous condition, this loci is not following the Mendelian laws. AA must give A blood group. BB must give B blood group. But that means both to form A antigen or B antigen. But because of the QT allele, it could not able to form any antigen and the individual shown as a O blood group. Understand? So it is different. This is called as a gene interaction. So gene interaction. So gene interaction. This loci we called as a epistatic loci. So this loci we called as a hypostatic loci. Hypostatic loci. So if there is no this one, absolutely it formed as a dominant and recessive. It forms antigens. The presence of homozygous footy allele, it prevent the antigen formation or working of this loci. So generally, very, very rare case, these footy alleles are present in an individual in homozygous condition. So 99.99% human beings, these footy alleles are only one dose, not the double dose. That's why whatever the genotypes present here, that genotype is expressed as an output. In rare exception, if an individual has two footy alleles, is antigen formation of this blood group loci modified or it's suppressed. So this is the first expression, epistatic genes. So epistatic locis, example Bombay blood group. So Bombay blood group, where individual genotype is AA or AO, BB, BO or AB, but output is O blood group. But output is O blood group. It should not be the case, but it is the case means it is an exception. Understand? So now second one. So sex linked one. So sex linked locis for say X dominant and X recessive case. So dominant and recessive come into being when two doses are there and these two doses are different in nature. So in blood group, if you take AO is A and A homozygous. A and O homozygous. So A and O heterozygous. Heterozygous. So in this case, none is dominant over the other. But in this case, A is stronger and dominating over the O, and the output is A blood group. Output is A blood group. Understand? So that means there must be a loci to be a dominant or recessive. It must be two alleles are present, and these two alleles are different in nature. In that case, dominant and recessive occurs. Understand? If an individual has only one chromosome and one chromosome has A or O, in this case, there is no other chromosome, other loci, other allele. So this is obviously expressed. This we call as a hemizygous. Hemizygous. So if you take the some of the diseases such as color blindness, so color blindness and hemophilia. So these are all, one is X dominant, another one is recessive. So in female case, two X chromosomes are there. So if one of the gene is color blind causing gene, another gene is non-color binding class gene. In this situation, what happened? So because it is recessive in nature, this is normal gene. So this is blindness causing gene. But this gene is recessive and this gene is dominant. So that individual never be a color blind. So never be a color blind, but carrier. So these are all the X. If you take the male, one is X chromosome, another one is Y chromosome. In this X chromosome, if small c is present, that is blindness causing gene, because lack of another gene, lack of another X chromosome, lack of another gene, so it is hemizygous. Hemi means only one, hemizygous. So no other thing to dominate here, even though single dose present is affected with color blindness. Is affected with the color blindness. So here also, hemophilia, it requires two doses. Here one dose is enough. Understand? So law of dominance and recessive, we will apply when a loci has two alleles. In the males, X chromosome is only one. Here, the loci means not the two alleles. One allele is enough. That means here law of dominance and recessive is missing when only one X chromosome present in the males, that is sex linked. So in the sex linked loci such as X dominant and recessive disorders such as color blindness. So color blindness or hemophilia. So no rule of dominant and recessive. No rule of dominant and recessive in males. Recessive in males. Why? Because only one X chromosome. So third one, sex influenced or sex limited. So sex limited characters. So do you ever observe female baldness? Female baldness. So male we can observe the baldness. So losing of the head hair, that is baldness. So here you can see in females, in the entire chromosomes, capital B is a gene or allele which causes the baldness. So small b is present on the same loci. Another allele, small b present on the same loci. So this is the loci. Loci has two alleles. These alleles might be a BB. Or another case, it might be a small b, small b. In another case, it might be a small b, capital B. Three combination can be possible on this loci. Understand? So for suppose capital B is a allele which causing the baldness. And the nature of this one is dominant. Nature of this one is dominant. Small b is non-baldness. Non-baldness gene or normal gene. The nature here it is, the nature is recessive gene. So recessive gene. So that means one is dominant, another one is recessive. And capital B is baldness causing gene. So that means in BB combination, baldness come. Capital B, small b combination, baldness come. Why? Because capital B is dominating over small b and capital B causing baldness. Okay. So small b, small b combination, no baldness, non-baldness. Non-baldness. But it should be same in the male and female. But here you can see when it comes to male, and female. So in this case, both male is baldness, female is baldness. In this case, male is baldness, female is not baldness. Here, both are not baldness. Understand? So that means heterozygous baldness in the female is a non-baldness. Heterozygous baldness in male is a bald giving. Bald. So bald, non-bald. So no bald. Why? Because this gene never causes any baldness. Small b. So that means here what happened? Here also it should be a baldness. So because female have estrogen and progesterone hormones are different hormones which are all not present in the males. So those hormones influencing this one and it suppress the emergence of baldness. Because lack of these hormones in the male, so one dose of bald causing gene dominating over the normal gene and it causes the baldness among males. So this is how females have less balded head, males have more balded head. Either in the homozygous case or heterozygous case, male became bald. But female have only homozygous case, they get the baldness. So this is the sex limited trait. So this is also, if it is the law of dominance to be worked out, this capital B should be a bald in the female. Okay, here also law of dominance and recessive, the expression of the trait is modified because of the female hormone. So in this case also, law of dominance and recessive failed in female case. That's why it is called as a sex linked. So sex limited character, that is baldness. So baldness in male, in heterozygous condition. in heterozygous. But female, no baldness. But female, no baldness. So irrespective of the gender, in heterozygous condition, if they are balded people, then the Mendelian laws are working fine. Okay, in male, the Mendelian laws are working fine. In female, these are all not working because it is influenced by the female hormones. So next one, fourth one, exception. So fourth one, if you see, so sex influenced. So sex influenced genes. So what do you mean by sex influenced genes? So the genes, the loci which is related to the milk secretion, which is present in the male as well as the female. But this milk secretion genes are active and working in female only. Okay, sex influence gene, milk secretion genes. So milk secretion gene, so functioned in female. Functioned in female. So any dominant and recessive associated with the female, it is expressed. So though this genes functioning is absent in the male, whether it is dominant or recessive, nothing to bother. That means in this milk secreting loci, so both alleles are same, both alleles are opposite. So then there is nothing happened in the male, but because that loci itself is not functioning in the male. But here, homozygous loci for milk secretion, heterozygous loci for milk secretion, that we can observe only in the female, not in the male, because that loci itself is against in the male. So here also law of dominance and recessive failed in the male because the loci itself is not working. Next, qualitative. So qualitative and polygenes. So qualitative and polygenes. So this is a separate question. This also not following the Mendelian laws. For suppose, so skin color. Skin color is a trait. So skin color is a trait, which is these three locus combinedly give skin color. Okay, skin color means melanin present in the body. If the melanin present in the body is like 10 grams, okay, so each loci contribute 1/3 of the melanin. So 1/3 of the melanin. That means the total melanin in the body is the sum of the three locis. Understand? Sum of the three locis. So here single loci, single output is failed. Multiple locis or multiple genes together produce one character that is called skin color. Understand? So this also influenced by the environment. So these three locis are perfectly producing the melanin, but the person is always staying in the colder environment. Then these genes failed to produce the melanin sufficiently. That means genes working. So first of all, multiple genes are causing single output, and these function also influenced by the external environment, that is hotness, coldness. So together, that means these locis never follow the law of dominance and recessive. So law of dominance and recessive not followed. Why? Because this is multiple loci, single output genes. Understand? So here you can see qualitative. Qualitative means you can measure. So additive effect. So quantitative. Okay, quantitative genes. Quantitative genes, each loci contributing equal effect into the total output of the character. So quantification of the each loci, how much quantity produced by the first loci, 1/3, second loci, 1/3, third loci, 1/3, then it became 100%. So it's not the qualitative, quantitative and polygenes. Quantitative locis, quantitative and polygenes, they are also not following the law of dominance and recessive. Example, skin color, height, weight. So though they are under the control of the genes, but external environment can modify that. Understand? So these genes also not following the Mendelian laws. And sixth one you can see, so lethal genes. So lethal genes. Next topic is this lethal genes only. So we'll in depth we'll discuss. Lethal genes also not following the Mendelian laws. And seventh one is genetic imprinting. So genetic imprinting genes. So this is a separate question. We will discuss today or tomorrow. Genetic imprinting. So genetic imprinting genes. So genetic imprinting genes also not follow the Mendelian law of dominance and recessive. So next one. So extra nuclear inheritance. So extra nuclear inheritance. So extra nuclear inheritance means any disease in the mitochondria. So this is nucleus. Any disease in the mitochondria generally passed. Mitochondria is always coming from the mother. So generally it passed mother to all children. So father never pass mitochondrial related disease to the children. Only mother pass. So here also Mendelian laws are never work out. So Mendelian laws are never work out. So yesterday we discussed about the extra nuclear inheritance. So nuclear means DNA and genes, those inheritance. Extra nuclear means outside the nucleus, we have that DNA in the form of mitochondria. So mitochondria also have the own DNA. Any change in the mitochondrial DNA that led to the negative products, that products, so not following the Mendelian laws. So these are all the areas you have tried. So epistatic loci, okay, epistatic loci, sex linked loci, and sex limited characters, sex influenced genes, quantitative and polygenes, lethal genes, genetic imprinting, extra nuclear inheritance. So this we discussed yesterday heavily, and these two things we will discuss in the upcoming classes. Okay. So this is all about the exceptions. So if you have any doubt in this concept, you can ask me, I will explain. So if you have any doubt in this concept, so any doubts are there, you can text me, I will explain one more time. First of all, whether a loci is producing single output or not, single character. Okay. And that loci follows law of dominance and recessive. This is the two things we will consider a loci is a Mendelian loci or not. Okay. So if a loci is never producing any output, if a loci is producing multiple outputs, if a loci is acting as a regulatory gene or interacting gene. Okay. So these are all the locis. They are not following the Mendelian. So Mendelian laws means whether the parental genes, parental trait. So taster is a parental trait. Non-taster is a parental trait. So these traits are again and again appearing in the next generation or not. Okay, on what rules a child might be a taster or non-taster? Understand? So if a child is a heterozygous, the child is taster. If a child is homozygous, okay, homozygous condition, small t, non-taster, capital T, taster. So that means we know clear rules about the trait inheritance, the successive generation. But here clear rules are not applicable because these functions are influenced by another mechanisms. So that's why these many genes, all these genes together, how many are present? So all these genes, these type of genes or these type of locis are nearly 19,000 locis. Nearly 19,000 locis have these characteristics. So blood group, if you take the blood group, I will explain. A child is born with A blood group. A child is born with A blood group. So what about is neonatal, infancy, childhood, okay, adolescence, adulthood, old age, and death? So can he change his blood group? So some days he resided in deserts. Sometimes he resided in the mountain areas in his lifespan. Sometimes he resided in the polar region. And finally he died. So means he had spent all the climates and all the phases. And he finally died. In any stage, his blood group is changed because of environment. It's not possible. It's not possible, right? So blood groups are never subject to change. Okay. So if you take a person, he born in Equatorial Guinea or Africa, so dark skin color, dark skin color. So due to family occupation, the family is moved towards the Sweden, or you take any polar country, Sweden, Denmark. Okay, so these countries generally present in the polar countries, and the rest of the after 50th year of this child birth in the Africa, their family migrated to the Finland, Sweden, Norway, which are all polar countries, and he grown in that countries. His skin color here is dark. Okay, dark skin color. Here light white spots appear. Light white spots appear. So dark to somewhat white. The pure dark is completely eradicated and whiteness gradually appears in his skin. That means here the skin color is a output. Skin color is a output. But these output in his lifetime can be changed when he changed the environment. If you take the some genes, 5-6 genes together controlling the person's weight, that means they can give weight up to 100 kgs. So the genes at full functional level, they can give the weight for an individual at 100 kgs. But he born in a poor family. The family is unable to nurture him or feed him properly. Then he stopped at the weight of 50 kgs or 60 kgs. That means though genes has the potential, but external things are required that fully potential realization. So these kind of genes we call as a polygenes. Polygenes never follow Mendelian laws. Understand? So where Mendelian laws means single output, and that output in your lifetime never changes. So those loci, those outputs, those characters only we call the Mendelian locis. So subject to change, multiple outputs, controlling genes, so epistatic genes, or these are all the genes are not following the Mendelian laws. Okay, every loci in our body following Mendelian law is a myth. Only one section of locis, one section of the genes, they follow Mendelian laws. Understand? Remaining genes are not Mendelian locis, not Mendelian genes. So one of the genes which never follow Mendelian laws are lethal genes. So lethal genes. So lethal, sublethal, semi-lethal. So lethal, sublethal, semi-lethal. So lethal genes, sublethal genes, semi-lethal genes. So pleiotropism. So pleiotropism means a loci producing the multiple products. Product 1, product 2, product 3, or so on. Multiple products produced by a single loci. Those locis we call as a pleiotropic locis, not the Mendelian locis. So lethal genes are, so for suppose if a person has Huntington disease, if a person has Huntington disease, so Huntington disease is caused by a one of the loci. It gives mental retardation, one character, muscle weakness, another character, some another symptom. That means loci is one, but how many characteristics it has given? Is that multiple characters or not? Are there multiple characters or not? So mental retardation is one type, muscular atrophy or muscular weakness is another character, some another symptom is associated with Huntington disease. So that means pleiotropic genes are, that means multiple output giving, multiple character giving, generally detrimental for the individual. Understand? Pleiotropic genes are generally causing the morbidity or mortality are negative for the growth of individual. So negative for the growth of an individual. So those kind of these genes we call as a lethal genes. So lethal genes means those loci are those genes which causing the morbidity, mortality, or any kind of infertility or sterility, or that means normal bodily functions are negatively impacted. That kind of genes we call as a lethal genes. And some cases this lethal genes produces ultimate result is death. Okay, that means some genetic disorders are very severe. So very severe means what? So that genetic disorder caused by a single loci. So it causes the failure of the brain functioning, heart functioning, okay, liver functioning, urinary tract functioning. Understand? So pancreas functioning failure. That means single loci causes the multiple phenotypic effect at multiple organs. So that means single gene, single loci, multiple outputs. That we call as a pleiotropic. When single gene has these much of effects or products, generally these are all death causing genes or morbidity causing genes. So that means lethal genes means a lethal gene is a gene or loci which produces morbidity, mortality, or sterility, or negatively growth and development of an individual. Those genes are lethal. The English dictionary meaning of lethal means dangerous, detrimental, death. So lethal gas, that means dangerous gas. Who inhale that gas, they will die immediately. That means the individuals who possess more lethal genes, quickly he dies out. Understand? So genetic disorder causing genes. Lethal genes are nothing but genetic disorder causing gene. Each disorder give multiple symptoms. These symptoms are negative impact on the bodily growth and development. The person quickly reaches to the death process. So those genes we call as the lethal genes. So again on the base of time of death. So time of death of an individual, the lethality varies. So the individual starts his life as a gamete. So ova, female gamete, sperm, male gamete. So when these two fertilized, individual comes into being. So when the individual born before itself he dies out, that means these ovas or sperms have defects in formation. They are unable to survive and fertilized. That kind of gametes we call as a gametic lethal. So the ova has to be transported into the uterus. Okay, so the sperm has to be swim in the liquid and it has to go to the fallopian tube to be fertilized. But all these activities of sperm or ova regulated by the genes present in ova and sperm. If they are unable to move, if they are unable to survive, that means the genes inside that ova and sperms are not functioning accordingly. So those genes we call as a gametic lethals. The gametes are unviable, unable to survive, unable to fertilize. So this is nothing but a absolute lethal. So absolute lethal means, so they cause the death of the individual before formation at the stage of gametes. So that we call as a absolute lethal. That also called as a meiotic drive. So meiotic drive or so meiotic drive. Understand? Segregation load. Segregation load. So that means the gametes are not properly segregated, not functioning healthy to fertilize, so that miscarriage or the cycles are wasted, the sperms are wasted, they never be realized at an individual human being. So that we call as a absolute lethal based on the time of death. The second one is some locis when the fertilization time, okay, embryo time, fetus time, neonatal time, okay, prenatal as well as infancy, but childhood time or puberty time, they starts to produce negative impacts, multiple negative impacts on the body, so that the individual dies. That means before reaching into the adolescent age or post-adolescent years to get the sexual activity, and to produce the to produce the children. So before producing the children, they should get married, then only they produce the children. Marriage means general 25 to 30 years or 20 years. That means these individuals, because of possession of some lethal genes, they dies out before marriage and before passing their genes to the next generation. When they produce the child, their genes are passed to next generation. Before getting married, a person dies means that we call as a sublethal. Understand? So absolute lethal means at gamete stage, individuals dies out. Sublethal means the individual dies out during the childhood or puberty ages, but not into a marriage age and producing the children. So these kind of example we can call as a sublethal. This we call as a sublethal. So sublethal causing the death of an individual during the late childhood or early puberty years. So example is epiloia disorder. So epiloia is a disease which, if a person has this disorder, which produces the multiple effects that led to the death of an individual before marriage or before reaching to the adolescent years. So third one is, so some of the genes, some of the loci, they produce the multiple effects, multiple negative effects, which result into a death at the age of 40 years. Semi-lethals, generally, semi-lethals is a loci or a genes. They cause multiple negative impacts on the body, which led to the death in late 30s or early 40s. Early 40s. That means by that time, they got married and they passed these genes to the next generation. So that example is like Huntington chorea. So Huntington chorea is a disease which phenotypically express symptoms, that is muscular, mental retardation, muscular dysfunction, and the finally death of an individual. So that means the symptoms, appearance, and death generally happen in the 40s, not in the before the 40 years, so that the genes are passed to the next generation. So this is the way lethal genes are classified. Lethal genes means they are dangerous genes which can produce multiple effects on the body. So these effects are negative in nature. The individual dies out. So absolute lethal means these loci never allow to realize an individual. Okay, sublethal means they allow individual to be born, but never allowed to be fertilized or grown into adult people. Semi-lethal means these lethal genes allow an individual to grow into adolescent, adulthood, and produce the children, but late in time they come into being in the symptoms and he dies out. So this is the lethal genes. So if a person died out means any genetic disorder which have multiple phenotypic effects, each one is a negative. So this is how you can see. So again, lethals are classified. Lethals are classified based on the time of death. This is the first classification. Lethal genes are further classified on the basis of nature of the loci. So lethals further classified. Lethal gene classification 2 based on the nature of the genes. So nature of the genes. So first one is autosomal or dominant lethals. So dominant lethal means single dose is enough for die. Single dose. Single dose. That means the loci has two alleles. One allele is a disease causing allele. Another allele is a normal. In that case also the individual dies out. So they're very, very dangerous. One dose is enough. In the recessive case, what happened? So both alleles should be a disease causing. If one of the alleles is disease causing, the individual became a carrier, but not the phenotypically that person never dies out. But in dominant, if one gene is disease causing, the person dies out. Here two genes must be disease causing, then only the person dies out. Understand? So dominant lethals are very severe lethals because single dose is enough to get the death. Examples are Huntington chorea, So Huntington chorea and epiloia. So these are all dominant diseases. So recessive lethals. So recessive lethals requires 2 copies. So these recessive lethals examples are hemophilia disorder. So hemophilia. So hemophilia is a recessive disease. Okay. So that means hemophilia impact in an individual only when it has 2 genes are hemophilia disease causing gene. If one gene is normal gene, another gene is hemophilia causing gene, the person never has symptoms and the person never get death. Okay. So third one is so incomplete dominance. So incomplete dominance. So incomplete dominance is a case of West Africa. In a case of West Africa, especially equatorial belt or Indian case also, India, Telangana, Andhra, Maharashtra, wherever forest cover is very high, malaria is very high. So malaria caused by a parasite, Plasmodium parasite. This parasite hosts are mosquitoes. So mosquitoes are heavily bred in the greenery areas. Understand? That means wherever forests are there, green cover is more. Where that malaria is endemic. Wherever forest cover is less, the malaria is very less. Okay. So in West Africa, especially equatorial region, the thick forest cover is there. Here malaria is present. So in these areas, the human beings have distinct hemoglobins. So this is the loci. So this is the loci which is responsible for formation of hemoglobin. Hemoglobin. So if this loci has AA, so the gene which cause the alleles which HBA, HBS alleles are there. So HBA, HBS alleles are there in West African people. So multiple alleles are there related to the hemoglobin, like A allele, B allele, O alleles are present in the ABO loci. So this hemoglobin producing locus are multiple alleles are there. One of the allele is HBA, another allele HBC, another allele is HBC. Okay, so HBS, multiple variants are there, multiple alleles are there, which causes multiple hemoglobins among the human beings. Understand? So here the people who possess the HBA allele. So HBA allele. So A hemoglobin comes into being. So A hemoglobin. If some of the people they possess HBS allele. So S hemoglobin present. S hemoglobin present. Okay. So now you can see A hemoglobin produces RBC cells into round in shape. Okay. So S hemoglobin produces RBC shape into sickled cell. Sickle. Okay. So now you can see. So this rounded one easy to travel in the blood capillaries. In blood capillaries, all are round, easy to travel. Understand? So if these are all sickled shape, so one is this one. If another one is knocking like this one, so then the clot occurs. Understand? Whenever blood travels into the veins, when multiple veins are there, so if they struck at these regions as a locking, so rounded one never lock each other, but this kind of sickled one they lock each other. So that kind of clogging occurs. RBC's clogging occurs. The blood has to apply more pressure to pump the blood. The heart has to apply more pressure to pump the blood because the blood clots are occur or clogation occur. This clogation led to the more pressure required to pump the blood for every cell in the body. Here, the movement of the RBCs are very free because everything is rounded. So no interlocking is possible. But this sickled RBCs are formed into locking system that led to the more pressure on the heart. So more pressure on heart. that led to the heart attacks. In West Africa people, whoever having both the HBS alleles, they have RBC cells, but their shape is lacking. They die in the 20s or before 30s, most of the people with a heart attack. Okay, these people are surviving well. Surviving well. So now you can see conditional lethal. We are discussing if a person has, that means this is the male person. Okay, so this is the female person. This female person might be died at the 30 years. Before on that, she got married and she produced a progeny. So the progeny has the combination of HbA and HbS. HbA and HbS. So now among all the RBCs in the blood valves, some are round, some are sickled. Some are round and some are sickled. So when compared to previous case, here half of the RBCs are round, half of the RBCs are sickled. So now lifespan of this person. Okay, so means when compared to this case, here heart should apply pressure, but this pressure is very less. So heart should apply more pressure to pump the blood. But when compared to second case, here the pressure is very less. That means these people will get the heart attack, but in their 50s or 60s. Lifespan is more in an individual which has the heterozygous condition. Understand? So now the scene is not at over. So we are talking about the region where malaria is endemic. Too much of malaria every year during the rainy season, or so in equatorial region everyday raining. Okay, malaria is everyday case. So the malaria plasmodium parasite, it is very, very fond of about the rounded RBCs. So if a person bit by mosquito which has the plasmodium parasite and the parasite enter into that individual, if the individual has all the RBCs are rounded in nature, so this parasite kills this rounded RBCs very highly. Understand? So the same individual which has, okay, but when compared to rounded RBCs and sickled RBCs, parasite kills rounded RBCs more, and this is least impacted by the malaria. This is least impacted by the malaria because that parasite, that much damage is not causing for the sickled RBCs because it might not be getting the grip over this sickled one, but rounded one it is easily getting the grip. Okay, so now you can see these people are dying out due to malaria. Okay, these people are dying out with heart attack. But here in this case, if a person bite by mosquito and these rounded RBCs were destroyed by the parasite, so some temporarily, some period, he can survive due to sickled RBCs support them. Okay, by this time, he can regenerate the rounded RBCs. Understand? So though he suffered by the malaria, rounded RBCs are comes into rescue to transport the oxygen. So the sickled RBCs survive and they transport the oxygen to every cell so that the individual survive. Even though rounded one destroy, sickled one help to survive. In this time period, he again regenerate the rounded RBCs. So this we call as a heterozygotic advantage. So heterozygotic advantage. In this malaria areas, an individual born with a heterozygous condition, HbA, HbS, is advantage than the homozygous condition, that is HbA, HbA or HbS, HbS. So incomplete dominance. So that means according to Mendel laws, if two different alleles are present, only one to be expressed. Okay, so in this blood group system, if AO is there, what is expressing? Only A. O is suppressing. But here A causing the rounded RBCs, S causing the sickled RBCs. Why means neither A nor S are dominate over each other. So it is incomplete dominance. If one of the thing is dominated over another one, either round to be born or sickle to be born, because both are unable to dominate each other. Both are functioned, both are produced different shapes. So this is incomplete dominance. In incomplete dominance also, especially in this hemoglobin example, this condition is a fatal. Okay, this condition also a fatal. Understand? So incomplete dominance. So incomplete dominance in the form of West Africa, hemoglobin. So hemoglobin. So HbA and HbS. So this is incomplete dominance. If one is completely dominant over the another one, all the RBCs should be either rounded shape or sickle shape. But each one is unable to dominate over the another one. It produces both types of RBCs, and that condition also causing the death. But these people survive more, but they ultimately die when compared to the another non-African individual. Non-African individual, if they live like 80 years, 19 years, these people die out like a 50 years or 60 years. So this is incomplete dominance. And the fourth lethal R is nothing but so erythroblastosis fetalis, conditional lethal. So conditional lethal example is erythro. So blastosis fetalis. So erythroblastosis fetalis. So this is the loci. Okay. So this is the loci chromosomal one pair. In the one pair, there is a RH factor. If both are RH plus gene, it produces RH and antigen D on the RBC cell surface. This we call as a RH positive people. Okay. So if both are RH minus, so it is unable to produce any antigen D on the RBC cell surface. This situation we call as a RH negative people. So presence of antigen D on the RBC cell surface we call as a RH plus people. Absence of antigen D we call as a RH negative people. So it was produced by the first chromosomal pair on one of the loci. Understand? So if one of the person is RH plus, one of the parent, another parent is RH plus, then child is RH plus. Okay. So one of the parent is RH minus, child is RH, another parent is RH minus, the child is RH minus. Understand? So one of the parent is RH plus, another parent is RH minus. RH plus is always dominating over the RH minus. The child is born with RH plus. So RH minus, in this case, RH plus is output. Why? Because it is dominating over RH minus. Understand? So this is the case. But here there is a problem. So in normal case, this is the case, there is no problem. Absolutely compatible marriage. So this is also a compatible marriage. But if father is RH plus, mother is RH minus, then they can get only one child. In this condition only, they can get only one child. How? So RH plus means it produces antigen D on the RBC cell surface. Understand? So mother's RBC cell surface has so father, see, this is the case, right? Father is RH plus, mother is RH minus. That means father's RBCs has antigen D. Okay, father's RBC cell surface has a protein we call as a antigen. That name is D. Okay, so mother's RBC cell surface has no antigen D. Understand? So when these two fertilized, okay, they got the mating and they produce sperm and ova. Okay, male sperm able to produce antigen D, female sperm unable to produce antigen D. Okay, so now the baby is growing inside a baby, inside the womb. Inside the womb, baby is growing. So what is the baby's combination? RH plus. So baby blood group is nothing but a RH plus. RH plus means baby's RBCs have antigen D. Baby's RBCs have antigen D. But the baby connected to the mother through placenta. So this placenta is used to circulate the blood. Okay, this blood carry the water, waste, nutrition. So between the mother's body and the fetus, the placenta role is exchanging waste, water, blood, and everything. In that blood, okay, in that blood, there is an important component of immunity. So T cells. So T cells role is, T cells are circulating in the blood. T cells role is identifying the foreign materials and destroying. So identifying the foreign materials and destroying. So when T cells of the mother, circulated into the fetus blood and mother's blood, exchanging each other for nutrition, waste, and everything. So when fetus T cells are circulating into the mother's T cells are entering into the fetus blood. So it identifies that fetus has RBCs, but it has antigen D. And mother's immunity system knows that it never has familiarity with the antigen D. Why? Because mother from the birth, she has no antigen on the RBC cell surface. Suddenly T cells come to the encountering of a RBCs which has antigen D. And they thought that there are foreign material in the mother's body. They start immediately producing the antibodies. So antibodies to destroy that RBCs which has antigen D. So those RBCs are babies. Okay, so that means the baby should grow into a 2 months or 3 months. After that, the blood forms into the baby body. That is, fetus generally circulated the blood by the time of 3rd month. 3rd month or 4th month onwards, the baby body, that is fetus body, has circulation of the blood. And that means mother's immunity system come to know that baby has foreign material in 4th month or 5th month. So and immediately it informed to the immunity system to produce to kill these cells. But that antibodies produced, and by the time they are taking action, the fetus to be grown into 8 month or 9 month. So 8 month and 9 month, and the fetus is safely delivered. So that means for the first birth, there is no problem. Okay, when second birth happened, so second fetus is growing. When first RBCs are born in the second fetus, by the time mother immunity system had already developed antibodies, the first RBC cells itself kill. That means second embryo or second fetus unable to produce RBC cell surface, so that the fetus would not able to grow and survive, and it terminates. That we call as a erythroblastosis. So that it is terminated in the form of omitting the blood, that is blastocyte. So erythroblastocyte. Erythroblastosis fetalis means the fetus terminated because of the blasting of RBC cells. That means in second time onwards, it's not possible her to conceive from the same father. Okay, same husband. Why? Because her immunity system immediately recognize and kill whatever the RBC which has antigen D. In the earlier case, it took the so much of time to generate the antibodies. But this time memory is already present for the antibodies for antigen D. Immediately they create the antibody and kills it. Already circulating antibodies are there. Whenever they encounter the next time antigen D RBCs, they kill immediately, so that now no blood formed into the fetus. Because lack of this blood, the fetus unable to receive the nutrition and everything, the fetus dies out. So it happens in the second pregnancy onwards. So that's why many people, they thought that they are conceiving, but the fetus is aborting in the third month or fourth month. The reason is this: if father is RH plus, mother is RH minus, in that case, only first pregnancy can be possible. Second pregnancy onwards, the fetus is automatically aborted, terminated, because lack of RBCs. So now, if father is RH plus, mother is RH plus, there is no problem for the second baby, third baby, because both bodies know about the antigen D. So antigen D, both bodies are familiar. If both are RH minus, both bodies not familiar towards the antigen D, then there is no case. If mother is RH plus, okay, father is RH minus, so these person unable to produce any antigen in the mother's body. If antigen is present, that is familiar. Not present, if foreign material is present, action would be taken. If foreign material is not present, no action would be taken. So in this case also, no problem for the pregnancies. If father is RH plus and mother is RH minus, in this case, second pregnancy onwards, it terminates. Understand? So among all these things, only in this condition, the problem arises. That's why we call it as a conditional lethal. So these loci only lethal when father has RH plus, mother has RH minus. Remaining conditions, these loci is not a death causing loci or morbidity, mortality causing loci. So this is how you have to categorize the lethal genes. So this question, heterozygotic advantage. So why in West Africa heterozygotes are more surviving than homozygous? Means one people dies out because of parasite problem, that is malaria. Another people dies out because of heart attack. So third condition, they survive more than these two people. So that's why we call as a heterozygotic advantage. In malaria endemic region, the human bodies, so human bodies responded to increasing the longevity or lifespan by producing the new hemoglobin, that is HbS. So possessing HbS in malaria area is a adaptative feature. Adaptative feature. But the same hemoglobin in another areas is a mortality cause. Understand? So this we can say in evolution process, why different products are given by the same loci. So different region, same loci gives different products. The reasons is like adaptability. That we will see in the evolutionary anthropology. So this is all about the lethals. So lethal genes are nothing but pleiotropic genes. So produces multiple negative effects. And finally death. So classified into two categories: absolute lethal, so semi-lethal, so sublethal and semi-lethal. So second category is dominant lethal, so recessive lethal, so incomplete dominant lethal, and conditional lethal. So this is on the basis of nature of the gene. Nature of gene or nature of Loki. So death time of the death. So death time. So this is the lethal classification. So tomorrow we'll discuss about genetic imprinting. So genetic imprinting. So genetic imprinting and polygenes. Polygenes. Difference between qualitative genes and quantitative genes. Qualitative genes and quantitative genes. So after that, we will finish the this kind of Mendelian genetics chapter. So if somebody asks you like, so you can write one term. So misuse of Mendelian genetics. So we'll discuss it tomorrow. Misuse of Mendelian genetics. Misuse of Mendelian information. Mendelian laws. So misuse of Mendelian laws. So what do you mean by misuse of Mendelian laws? So very simple, I am telling. Few males possessed a low key. Few males possessed a low key, and this low key giving better fitness for the children. Better fitness for the children and better sexual ability for the individual. This one. So now fitness for the progeny, or this loci has better sexual fitness for the males who possess this loci. So this is the case. And in a society, if these people are present like few in number and non-possessor of this loci are more in number, more males are there and females are there. And when we come to know that this loci is adoptive advantage for the himself as well as the children, all the females wanted to marry these only. Understand? So that means imbalance occur. The selection process, mating patterns, everything changes. Eugenics, I told you, right? So eugenics means everybody wanted a partner with better quality genes. Then obviously remaining people will be neglected from the selection process. Understand? So nature operates on the diversity. If everybody wanted a few selective locis, few selective genes, then what about that remaining diversity? That is eradicated. That is eradicated. So why nature maintaining the diversity means in an environment, some people are fit, other people might not be fit, but environment may not be constant forever. If environment changes, whatever the fit today, they will become unfit. Whatever the unfit, today they became the fit. And we artificially, because of knowledge, we are only promoting certain traits and we are eliminating the remaining diversities. In this case, when environment change, today fit traits are became negative and all the human race dies out. So that's why according to religion or according to science, no trait to be propagated as a good trait. It is against the human population. But when we have better knowledge, we are artificially applying this knowledge in our everyday life. That is problem. So tomorrow I will clearly explain. So most probably 1 or 2 classes we will finish all the related topics. Then we will move to the next chapter. So if you have any doubts, you can let me know. So that is evolutionary anthropology. It takes time to build basics in evolutionary anthropology. So if you have any doubts, you can let me know, or else we will end. Any doubts are there? Okay.